All right, so welcome to our 11:30 session. I got my eyes looking right here. I don't know which number it is, session 4, I guess, of day 2. Dana Pasquale is working with our, many of you know her. We've emailed with her. She's been a stalwart supporter of social networks and health for years now, and has recently taken a position as an assistant professor in population health at Duke. So congratulations to Dana.
Thank you.
And so Dana, we'll turn the floor over to yours. It's all you.
Okay, so I do have some slides and I hope that everyone can see them. Thumbs up. Yep, okay, cool. So it seems like, it'd be nice to do a short intro, but I encourage anyone to stop me along the way as we're going. So I just wanted to talk about recreating networks using epidemiological and phylogenetic information. So this is just going to be a quick intro, a subset of the full slide deck that's posted online. But really all this boils down to is that it's another way to define nodes and edges using pathogen gene sequence analysis and some information about contact networks. So it's going to sound a lot more complicated than it really is. So we've probably all seen a tree of life image where different animals are represented in the evolutionary tree. So historically, we used morphological characteristics to classify organisms like, do they have internal or exoskeletons? Do they have web feet and wings and things like that? But now we tend to use genetic similarity. It's an advancement of science. And then when you look at the genomes of two different organisms and figure out how genetically similar they are, you can build a similar tree. So we take that information, that genetic similarity from the genome, and we put it into a tree that's now called a phylogenetic tree. And that's a visual representation of the amount of genetic similarity between different organisms. And in this case, these are specifically African antelopes. They're adorable. To me, they all look exactly alike. And so the next question is, how do we sort this out? So recall that our genetic material is made up of A's, T's, C's, and G's. Those are the nucleotides. And those 4 nucleotides make up our genetic code. And then a set of the nucleotides, as they all are together in order, as they were sampled from an organism, is called a sequence. And if you can obtain sequences from multiple organisms that more or less start in the same place in the genome, then you can line them up in such a way that there's the least amount of penalty in the change in nucleotide from one sequence to another. And then you can assign a similarity score based on genetic similarity, and that's called an alignment, and that's what's shown here. And the method of similarity chosen depends on the organism and your goals. So you can then look at the similarity of sequences to build a tree where the branches are based on divergence among the sequences using maximum likelihood or Bayesian processes. It's a statistical method. And then using the alignment, you can create the phylogenetic tree, which is, again, the visual representation of the amount of genetic similarity of different organisms, similar to the way that a tree of life measures morphological similarity. And then each makes inferences about the common ancestors that are unsampled. So the branch points, those nodes are the inferred common ancestors, but they're unsampled, which is why they're called inferred. And in these diagrams, it's the amount of lateral distance that tells you the amount of genetic divergence. So the vertical distance is meaningless. You can flip around the node points just for visual, ease of visual. You can stretch them out. It really doesn't matter. And then in any tree, you want an appropriately chosen outgroup, and that should be distantly related enough that you can see the amount of similarity, but not so far apart that everything looks like one big cluster. Like I wouldn't want to use a goldfish here, because then the amount of similarity in the antelopes, they would be so constrained that I wouldn't be able to get a sense of the difference. So we can also do this with pathogen gene sequences collected from different people. So if someone has an infection, that infection also has genetic material. We can extract that infection, get the genetic sequence, and then do the same thing. So the slide that I showed earlier that I'm showing again is actually publicly available HIV gene sequences collected from different people over time. And then the HIV gene sequences are aligned so that people can be grouped together by inferred transmission chains by inferred transmission clusters. So a measure of genetic divergence, which can either be a percentage or absolute, and then can be and should be weighted differently across different parts of the genome, is calculated to determine the genetic similarity. And then that is used to create inferred transmission clusters. So this is a phylogenetic tree that's a visual representation of the HIV infections of 804 people who were sampled in North Carolina over a 17-year period. So these are not publicly available. Each terminal branch, so this is a circular tree, not a linear one like we showed. So each spike is a terminal node, that's one person. And then the sequences, the 4 biggest clusters are picked out here in color. So we can use the sequences to group people together by similarity, and then we put them in a cluster. Those are inferred transmission clusters, which are in people that have very similar HIV infection. And we can do this because the virus mutates over time between people as a result of selective pressure. So in this tree, I said the samples were collected in North Carolina from 1997 to 2014. There are some limitations here. First is that we can't infer direct first-degree partnerships. So missing data is one of the biggest limitations of phylogenetic analysis. So if I have 2 people that are in a cluster together, I can't say that they were definitely partners. There could have been an intervening person who wasn't sampled. I also can't infer the directionality of transmission. I've got 2 people together. When I'm doing a population-based study like this, I can't say that person A transmitted to person B. And again, there can always be a person C in the middle there. Incomplete sequence coverage, not everyone who's infected is sampled, and then not every person who's sampled makes it into your data set. And then it's a fairly new field of study. There's really not a consensus on the relative percent difference that you should use to put people in a cluster together. A lot of it depends on your data. I'll talk about study design at the end, your question, and your study design. So the field of molecular epidemiology aims to use these kinds of data. And a couple of key places are to understand transmission networks, to confirm outbreaks, or to guide intervention. So in order to best apply pathogen genetic data to understand disease transmission networks, and that's specifically networks or clusters of people with like infection to understand transmission with some amount of granularity, the mutation rate of the pathogen needs to be in such a place that it doesn't mutate so fast that you can't put people together in a cluster, and it doesn't mutate so slowly that you end up clumping everyone together. So this has been done, I don't know if anyone's been paying attention for the last 2.5 years, but we've done this a lot with the SARS-CoV-2 pandemic. So we've been taking sequences from people around the world, and I'll show that momentarily, to get a sense of how the pathogen is moving around the globe. This type of information was used to recreate the 2014 Ebola outbreak in Sierra Leone. And then I'll also show one of my favorite papers, which is looking at a hospital outbreak with a multi-drug resistant bacterium in a burn unit. So in this analysis, this is the COVID-19 pandemic from NextStrain. You can see that there are only 3,000 sequences here. That's the red oval in the top left. There are about 3,100 genomes here, but at the time that I took this, there were 504 million cases worldwide. So this is just a very, very small subset of what's happening. But what they're doing, and this is the phylogenetic tree in the top right with the variants and subvariants by color, is they're looking at pie charts to see where these variants were detected around the world, and then making some kind of inference about how these different parts of the globe were linked together based on genetic similarity. But in limited cases, and so that's not really a person-to-person network, it's a geographical network in the last slide. But in limited cases, with the right data and under the right conditions, you can reconstruct person-to-person networks. And I highly recommend this paper here if anyone is interested in this kind of analysis. So the investigators use molecular epidemiology to understand the transmission of Acinetobacter, which is in this case, it was multi-drug resistant. It's a bacterium that occurred over 3 years in patients in a burn unit. So it was a hospital outbreak. using the sequence of the pathogen, really detailed information about the locations of the patients and the care teams of the patients, and then some environmental sampling, they combine the molecular and epidemiological information to create a network, and then refined it using Bayesian methods to estimate a final putative directed person-to-person transmission network. So the interesting thing too about this analysis is that they actually, you've got two components here. They actually identified 3 distinct outbreaks. So the first outbreak starts with that dark red node AO3, and the nodes are colored by the date of infection. And it moves along toward the end, the last 3 nodes in that, starting with orange A15 and then A25 and A36, those were determined to be a second outbreak. So the people at the last 3 people in that component were determined to be infected with the bacterium that caused the first outbreak that recombined with a bacterium that was circulating in the hospital. They did some environmental sampling at the same time. So it was determined to be a second outbreak. And then the third outbreak in the component at the bottom was a completely different subtype of Acinetobacter. So they did some environmental cleanup and were able to stop the outbreak. And then phylogenetics can also be used to confirm that an outbreak occurred. So the idea of contact tracing, we're probably all familiar with given the SARS-CoV-2 outbreak, is a strategy for infection containment that involves public health personnel in interviewing one case and then tracing that person's contacts to test them as well, and then figuring out how to locate and test everyone else who might be at elevated risk of infection due to contact. So in the course of contact tracing HIV in Western North Carolina, the North Carolina Department of Health and Human Services observed a surge in acute HIV infection in the western part of the state. Acute HIV, the period of acute HIV is really only about a month long. Most people are diagnosed after that point in time, but people with acute HIV are highly infectious because their viral load is going up a lot because their immune system hasn't kicked in yet to tamp down the virus. So people with acute HIV are highly infectious to somebody else, another sexual partner. So the Department of Health and Human Services was concerned that there might be an outbreak occurring from one highly infectious person serially infecting someone else who then serially infects the next person. On the other hand, case finding during acute HIV is really difficult, and it's a win for public health when you can diagnose someone really close to the time of their infection during that period of acute HIV. So they weren't sure if we had an outbreak of serial acute transmissions or better case finding. So they asked Ann Dennis, a physician at UNC, and I to assist. So we compared the contact tracing, which are the epidemiological links between cases and their contacts, with their HIV transmission clusters. This is a multiplex network. The black lines are relationships that were discussed or disclosed during contact tracing, and then the dotted green lines are the phylogenetic links. So anyone who has a green box, any of the nodes with a green box were nodes that had an HIV sequence for us to analyze. So we were able to determine that this wasn't actually an outbreak from one highly infectious person to another, because if they were, we would have seen that all of the components that had a bright green person would have been clustered together, but they weren't. It was actually better case finding. You can see that even with the genetic information, most of the people with bright green boxes around them weren't connected to each other. So it was better case finding. So this is actually an example of when molecular epidemiology was used to confirm that there was not an outbreak. Molecular epidemiology can also be used to guide interventions. So the same physician at UNC is using near real-time analysis of phylogenetic clusters to determine where contact tracing should be deployed. The idea is that if the cluster keeps growing, it means that transmission is still occurring from someone that's been diagnosed onward. And that's where you need to deploy that ring containment strategy of contact tracing. And then there are a few hospital consortiums which are tracking multi-drug resistant organisms among inpatients so that control measures can be implemented when necessary. So to implement these types of analyses, as I said earlier, it really is just another way to define your nodes and edges. So the applications are broad. It's a relatively new area of research. So applications are being developed, especially as computing gets faster and big data is easier to use. So there are the typical study design issues, the sampling frame, whether the population is appropriate, but in this case, you have to worry about both the host population and the pathogen. And then whether the pathogen is appropriate is an important question. So some pathogens hardly evolve and thus aren't really good candidates for recreating a transmission chain because you don't see enough differentiation from one person to another between hosts. And then in some cases, you may be able to adjust your goals to find clusters. And this is the kind of thing that's done when investigating like a foodborne outbreak. So you've got someone that has E. coli or salmonella, you can type everyone in the country, realize that they have the same type and that they all ate the same kind of lettuce, go back to the lettuce processing plant and then find the pathogen there. And then some pathogens mutate so quickly that you really can't match from one person to another. And then on top of that, the choice of gene also makes a difference. So you have to choose a part of the genome that is varying. So some parts of the genome are under selective pressure. So this is the HIV genome. The one in pink, the envelope gene, is under constant selective pressure from the host immune system. So it does vary. Polymerase in orange is the target for most HIV drugs. So it's under selective pressure if the person is treated. So those are the commonly used targets for any kind of analysis like this. So this is a network that I'm working on right now. It's an example of what happens if you don't choose a part of the genome that varies. It's a bacterium, Klebsiella. The bacteria have a core genome, but they also have the ability to pick up or drop extra accessory genes that encode for resistance. So the part of the genome that varies is not something that the bacterium even needs. They can drop this extra bit of genetic material whenever they don't need it because it's costly to replicate. So when there isn't selective pressure that selects for resistance, it's not something that's really there. On the other hand, there is a core part of the genome that doesn't really vary much at all. And so the color of the edge here is by time of diagnosis. Each of the nodes is an individual inpatient in a hospital. And you can see that many of the people on the left by the edge color were all in the same facility at the same time, all diagnosed around the same time, and have really similar infection. So the likelihood of determining a transmission chain is low using only the core genome, given the lack of pathogen evolution in the core genome. And so back to here, just like a network, there needs to be sufficient coverage in one area of time or space for connections to be observed such that you get a sense of the big picture. Since the pathogens evolve, the coverage by time or space is important. Otherwise, you lose potential edges if they're defined by the pathogen. And then some pathogens are better studied than others, in which case the mutation rate across different parts of the genome might already be known. So you can still draw an inference over a longer time period if you're using that mutation rate. So statisticians always like to say that you should involve them at the time of the proposal, not once you have already collected the data. So in these types of analyses, you should also add a bioinformatician or a phylogenetics methodologist to the early consult set. And then also make sure that that person is versed in your organism of interest, since there's a lot of difference in the methods for a rapidly mutating virus versus a more complex bacterium. So to define the edges, there are a couple of different things to think about. The first is the relative contribution of information from phylogenetics and epidemiology. So I'll show some work at the end where the molecular information is treated as the gold standard because the epidemiological information is poorly observed. But then for things like the Klebsiella network that I just showed, the bacterium that doesn't change, we definitely want to use a lot of the epidemiological information to refine the network because the molecular information isn't very informative. It's better to say that a link didn't occur than to say that a transmission actually happened. So of course, like anything else, you need to know the limitations of the molecular and epidemiological network data. Both tend to suffer from missing data as an important limitation. And then the genetic distance cutoff is going to be a function of the amount of observation you can make based on your funding, your data, your population, and the organism. And then genetic distance can be either an absolute or a relative measure. It can be pairwise from one person to another, one node to another, or it can be against a consensus, against a model sequence. And then one more note about the genetic distance is that it's basically a function of time or transmission. So the pathogens are subject to selector pressure from their environment, which could be other microbes if they're free living. Otherwise, it's the host immune system or even treatments such as pharmaceuticals. So looking at the research question, the pathogen, the population, and the ability to observe will dictate whether transmission clusters, which are 2 or more people with like infection, or a specific transmission chain can be reconstructed. So if in this study, which was a really important paper in the field of HIV, the investigators followed almost 1,800 couples where one person had HIV and one person didn't. Then they randomized half the couples to be receiving HIV treatment for the HIV infected person immediately after diagnosis. And then the others followed what was standard of care at the time, which is you didn't take a treatment until you started to have some kind of clinical reason to. So it was a progression of HIV on the way toward AIDS. And in this study, they were able to sample the person who was HIV negative in the couple repeatedly. So there was only a set time period where the person could have, any of the people who did develop HIV, who did acquire HIV, could have been infected. And they confirmed with phylogenetics afterward in the very small number of couples where the HIV negative person did acquire HIV. They used phylogenetics afterward to see if the strain that the person newly acquired matched their main partner that was in the study with them. And because the couples were observed regularly, they used a really small genetic distance cutoff of 1.5% difference across the gene that they used between the 2 different couples, which is a very, very small amount, but they could do it because they were observing the couples in near real time, and they would know that the pathogen hadn't had time to mutate away from the donor strain. So this was a case where the investigators were able to recreate transmission chains. So to talk about choosing a research question, you need to think about whether you're retrospectively assessing transmission, if you're aiming to create a network, whether you're estimating evolution and travel of a pathogen or the organism of interest. And then those kinds of things are what will guide the choice of genetic distance cutoff within the parameters of the organism, the pathogen, and the gene. So then with any research project, you'll use that as the basis for defining your nodes and edges. So the data can be expensive to collect as there are sampling and sequencing costs, and there are resources where the data are already available, 2 of which are listed here for secondary data. They're repositories for genetic information. Funded researchers agree to deposit some or all of what's sequenced for their studies into these databases to become publicly available, exactly the same idea as IdeaNet and people agreeing to make their network data publicly available. There is a lot of information in these databases, but you do get what you get, and there are very limited bits of information about the sources of the sequences. So then just like any other network, you've got your nodes, which are defined a priori, as are your edges, and then the edges can be weighted or unweighted. So here's the Klebsiella network again, but the edges are weighted by transmission probability, which is scaled in color, which here is a function of absolute genetic distance between every pair of nodes. And then also here, solid lines were people that were co-present, and dotted lines were people that were not ever observed to be co-present in the same facility. So the data set for this is just like a network data set. It's an n by n matrix. They have weighted edges that have a transmission probability assigned based on the genetic distance cutoff, and then also the amount of overlap any patients had in the same healthcare facility. And so that went a little bit longer than I meant to, but I do have my email address here if anyone has any questions.
Awesome. Thank you so much. I don't know any no problem at all with it being a little bit long. You know, thank you. We have a little bit of time for questions. I don't see if Jeff is here. Jeff, are you here? We might have a lot of time. So, pragmatically, can somebody who's like not been trained in genetics jump into this kind of work, or is it best to like, you know, bring you in as a collaborator to do it?
So I would definitely make a plug for having, you know, a bioinformatician or something. I mean, you can, you know, very much in the same way that someone who has relational data could get into network analysis. But, you know, the entire idea behind IdeaNet is that, you know, there are a lot of pitfalls. There are a lot of easy mistakes to make. It can be done. There are some good trainings online that will give you the basics, but especially some of the organisms can be complex, and you do have to have a little bit of understanding of the biology in addition to having some understanding of the networks and the relationships between people.
And has your work on this convinced you that it was really nice to see in the one case that the increase in contact tracing actually seemed to, or the genetic sort of markers ended up sort of saying that what we're doing in contact tracing was right. How often is that the case, or is it that we're so essentially, if you were a public health place and had to invest in one of these methods or the other, which one is better?
That's a good question, and I think it really makes a difference. So the physician at UNC, Anne, she's done really good work trying to direct where contact tracing should happen. So, you know, public health is a story of never enough resources, never enough people, never enough time, never enough money. But on the other hand, you really do suffer from a lot of problems if you're not doing that, because in order to have a sequence, someone needs to get HIV, then they need to be diagnosed, then they need to make it to their first care visit, because that's where they get their sequence. So when you have high risk populations like people who are intravenous drug users, you're going to fall right out of that before you even make it to that point. So that's where you need sociologists, right? And social network analysis to say these people are missing, or we're seeing something here that looks like we should have more edges than we're observing. I really do think that both methods tend to have strangely the same limitations, which is that they're both retrospective. They both suffer from missing data, and they're both really limited in what one can say. I think that together they work really well.
Awesome. Great. Any other questions for Dana?
Thanks, Jim. I appreciated yours.
No, thank you. This was fun. We should talk every once in a while.
I would love that. Yeah.
All right. I haven't. Jimmy, good to see you. I see you popped in. I haven't seen Jeff Smith or Nora. I've received an email from him yet this morning. But presumably we're going to do a session on missing data and imputation here in about 3 minutes, according to my clock. So let's take a break, and hopefully Jim will jump in, or Jeff will jump in, and if not, I will, take a crack at it, I guess. All right, see y'all in a minute.